Hello, welcome to the lesson on figures and tables. Let's take a look at where this lesson fits into our project management approach of writing your manuscript. We are still in the planning phase. I would consider making figures and tables part of the pre-writing process. So this lesson on figures and tables will conclude the planning phase. Then we'll be ready to move on into executing phase. This is one of the shorter lessons. However, that doesn't mean that there's less to do. It depends on how much you have already done with your figures and tables, and how many figures and tables you have and how complicated they are. So although it's going to be a short video, it's still going to be a lot of work for you this week. The learning outcomes are as such. At the conclusion of this lesson, you should be able to determine when to use a table, when to use a figure, or when to use a photograph or a diagram or some other kind of figure. And you should be able to explain the concepts for figure design and presentation. Now, why do we have figures and tables? Let's step back and think about that. Figures and tables are how you present your processed data. So you collected raw data and then you processed it by analyzing it with whatever method is appropriate for your field and for the type of experiments you did. And now in the figures and tables, you're going to present that processed data. So it might be means, plus or minus standard deviations. In some cases, you might be presenting the raw data that has a curve fit to it. And there's a lot of different ways that it can be presented. The whole point of figures and tables is to show the readers the outcomes of your experiments that you're doing to fill the knowledge gap and answer the research question. So how do you decide how to present your results? Are you going to use a figure? Are you going to use a table? Or maybe you'll just put it into the text. When you have only a few data points, let's say 4 or fewer, it doesn't make a lot of sense to make a table or a figure for that small amount of data. And so in that case, it's best to just put it into the text in a sentence in your results paragraph. A figure is going to be the best choice most of the time because figures are a lot more visual and they're easier to understand quickly, and they allow you to see trends and relationships more easily than a table does, where a table has a lot of numbers and it's more difficult to read. If you can use a figure, do that. When you need to use a table would be when you have a mixture of data types that would take too many figures to present. And so maybe it's more compact in a table. Another time you might want to use a table is when you need your readers to see precise numbers. So if the exact number is more important than the trends, for example, then a table would be the best choice. So let's talk a little bit about table design. When you design your tables, keep the reader in mind. This is always going to be something we come back to in this course, is keeping the reader in mind. What you want to do is make the table easy for the reader to follow. One way to do that is to keep the structure as simple as possible. Additionally, you should place familiar information on the left and new and important information on the right. For example, times or dates would be in the left-hand column, and then the numbers that you collected at those times or dates would be in the right side, to the right side of that first column. Also think about the table titles. So these are kind of equivalent to the captions for figures, but table titles are a little bit different because you don't have an extensive legend for tables. You have basically a descriptive title. Additionally, think about what you're going to put in the columns and what you're going to put in the rows. So you have headings for the columns and you have headings for the rows. It's easier for readers if you put the dependent variables in the columns and the independent variables in the rows. I think this goes along with placing the familiar information on the left. Now, when we think about figures, there are a lot of different types of figures, and I feel like it's out of the scope of this course to go into a lot of details about types of figures and how to make those types of figures. So I'd like to just focus on an awareness of them, and then I think you should get advice from your colleagues and senior scientists in your group about specific figures that you need for your paper. But in general, the different types of figures that you might choose to use are shown in this slide. So there are photographs. Use those when you need to show specific samples, microscopic images, aerial images, kinds of things that you can only capture in an image. They're very specific to your study. Drawings and diagrams can be very useful when you need to show flowcharts or decision trees, show how you built something or how a pipeline was constructed. Drawings and diagrams are great for that. Line graphs are probably the most common type of figure, and they're used to show dynamic relationships over time, concentrations over temperatures, different changing variables that might affect whatever is being measured. So that's what I mean by dynamic relationships. Scatter plots are great when you need to show relationships between two variables. So maybe you're measuring height versus weight, and you would have one data point for each height versus weight relationship for all of the subjects that you're measuring. And so you could plot that as a scatter plot, and you might fit a curve to that or you might not. Bar graphs are great when you have groupings of treatments, if you have different genotypes that you're comparing, or those kinds of situations. Pie charts are used occasionally. I don't see them used a lot, but they are handy when you're dividing the whole group of results into percentage categories. Box plots are great. They're kind of like a bar graph, but they have a lot more information in them, and they show the variation. And they show variation that can't be captured in traditional bar graphs, because not only do they show the mean, they also show the range, and they show outliers. They have the whiskers that extend beyond the boxes that have a lot of additional information. And then there are also principal components, figures that show how certain treatments or genotypes cluster together. So these are just some examples of figure types that you might use. You might use something that's not even on this chart. As you are making your figures or tables, keeping the reader in mind, I think it's important that you think about the following points. What is the primary outcome, in other words, the result, from this figure or table? So if you're going to make a figure and you're thinking about which type of figure to make, it's useful to look at this question. And think about why you did that experiment, why you collected that data. And that's going to help you decide what's the best way to present it. Secondly, after you've made your figure or table, or while you're making your figure and table, think about if you're presenting the data in a way that allows the reader to quickly see the results. So in other words, are you presenting the data in a way that makes it easy for the reader? Now at the same time, you don't want to cross the line to where you're leading the reader to a certain conclusion. So think about this question: is my presentation unbiased? Make sure your presentation is unbiased and it is not misleading. We're not setting out to reach a foregone conclusion. We're trying to find out the truth about nature and natural phenomenon. So we need to present the data in a non-biased way, and at the same time in a way that it's easy for the reader to see the outcome of the experiments. So for example, if you're making a bar graph or a line graph and you're comparing two different treatments, think about where you start and stop the scale. So should you start the scale at 0? Let's say if your numbers are 900 and 950, should you start the scale at 0 or should you start the scale at 850? If you present it as 850 to 1000, that could be misleading because it's going to make the distance between those two lines much greater than they would be if you started the scale at 0 and went up to 1000. So think very carefully about how you design the figures for that purpose.